Welcome to our webinar. As you know, the reason we do this is to really help you in your mission for treating brain injury. It's a big mission, and we're hoping these webinars will help. We have this series that contains tutorials, cases, and examples, and we also, in some of them, provide the data and tools you can use to work out some of the examples presented in the webinars. We also have starter grants that are in-kind grants using our time to help you with an idea or a seed project. And we actually have a couple of those ongoing now, and we're really enjoying helping people get their projects started. So contact Craig at moberganalytics.com if you have an idea and you want to run it by us. We're always happy to help. Please provide us feedback. We're always looking for new topics for the webinar. So if you want to speak, let us know. Or if you have suggestions for others, please contact Craig. He would be the best person for that. This is a list of the webinars that we have recorded, and they are posted on our website. We're getting quite an extensive list of these. We have we have like a core group that is here on pretty much every live webinar we do. But then the replays of this are amazing. We have a huge following of people that come in after the recording and listen to it. If any of you signed up for the webinar, and if it has assets associated with it, you should get an email that will allow you to go into our portal and to use those assets. As most of you know, this is a Zoom webinar, and the questions, if you have them, will be done through the Q&A button on your screen. And please feel free to send questions to us while the talk's going on, and we'll also give you a little chance after the end to submit your questions. So, without anything further, let me introduce you to a good friend of mine, and we'd like to welcome Jana Kainerstorfer, who's a professor and associate department head in biomedical engineering at Carnegie Mellon University. Her lab's research is focused on developing non-invasive optical imaging methods for disease detection and treatment monitoring, with an emphasis on diffuse optical imaging. Her research focuses on clinical translation for monitoring and assessing cerebral health. She's a world-renowned expert in biomedical optics. She serves as conference chair served as a conference chair for the recent Photonics West Clinical and Translational Neurophotonics Conference in San Francisco. She's the associate editor for the Journal of Biomedical Optics, and she's a senior member of OPTICA and a fellow of SPIE. Today's talk is entitled Body-Brain Behavior, Non-invasive Neurotechnologies for Peak Performance and Cerebral Health. And I will turn it over to Jana at this point. Thank you.
Thanks, Nick. Thanks for the introduction. Let me share my slides. My computer wakes up. Okay. Thanks, Dick, and all your team for the invitation. It's great to be here. And like Dick said, I'm a professor at Carnegie Mellon, and pretty much everything we do is focused on optics and optical imaging, optical sensing of the brain. And what I really want to talk about today is some of the most recent work we have done, as well as kind of a summary of the last decade at this point. Based on my affiliations, you kind of can tell where I'm coming from. I'm an academic. I am affiliated with biomedical engineering, so we build tools, and as well as with the Neuroscience Institute, so very much focused on neuroscience and so on. So as Dick said, we focus in the lab on optical imaging, diffuse optics. I like to say to our incoming undergraduate students, I just say what we do is fancy pulse oximetry and wearables, and we use a whole lot of wearables. And so pulse oximeters and Fitbits and Apple Watches are all very good in detecting respiratory changes, pulse oximetry, cardiac related issues. But those optical sensors, I want to take the opportunity here and talk a little bit about what is diffuse optics and the nearest devices that are available in the clinic, and kind of point out that the methods that we have in the university settings are typically more advanced than what you have access to in a clinical setting. But no matter how it's built, no matter how we use it, everything, including a pulse oximeter, is based on this idea that light gets absorbed differently depending on the wavelengths. We usually use near-infrared light. That's what NIR stands for, near-infrared spectroscopy. And the benefit of near-infrared light is that it goes all the way through the skin, through the bone, interacts with the brain, and we can build NIRS devices for brain sensing. And if we scale this up, we can do 3D reconstruction, and I'll show you some results on that, on how we can do some functional monitoring of different brain regions. And so I have worked with a lot of clinicians throughout the years, and pretty much always the question I'm getting is like, how is it different from clinical devices? And just as a comparison, there's a lot of really good NIRS devices clinically available. So that's the one on the right-hand side as an example. And those are great, but they don't necessarily have the temporal resolution or spatial resolution that we sometimes want for a research setting when we develop the next tools. Now, from a research perspective, what our instruments look like is something like that. It's a hospital card with a computer. This box on top here, I'll talk about, that's one that we build ourselves. It's often very clunky because we're academics. We somehow get optical fibers or LEDs on the head and I measure things. But there's a lot of advancement nowadays that builds research-grade devices. NearSense is a company that focuses on that. Artinas is another one, really focused on how do we build a near sensor that goes on just on the forehead, for instance, but battery operated and wearable. And so while those are just pictures, I hope I will convince you at the end of the talk that some of the research type grade devices are very useful and the next big thing for clinical translation. And as I said, if we scale this up, there is NIREX has really great devices out there that if you put many optical sources on the head, you can actually do mapping of functional activation. But as I said, I do want to talk a little bit about what is a NIRS device and what are the differences and what are the benefits of some. So typically in the optics community, we differentiate between what is called time domain, frequency domain, and continuous wave. Every one of those has the same origin of functionality, which is you take some sort of light source, shine it onto the forehead, light goes through skin, bone, interacts with brain, comes back out again. Now, the difference though between them is in time domain, we send a pulse of light in there and then measure when does the light come back out again. So you need really sensitive detectors for that. We're talking about nanoseconds, delay between one area and the other. Frequency domain similarly. Continuous wave, that's your classical nearest device in the clinic. That just means you send light in and you ask how much is coming back out again. So that's no different than a pulse oximeter, just a little bit better detectors. And so we can differentiate different depths inside the brain, depending on if the signal has traveled only a few nanoseconds, or if it has traveled deeper, then it will take longer for it to come back out again. And then continuous wave NIRS, we just change how far is the source and detector away from each other on the forehead. And so, but inherently the difference between the, let's say, more complex systems on the left versus the right ones is that classically all of the continuous wave clinical or research devices don't measure absolute numbers. We can quantify how does oxygenated and deoxygenated hemoglobin change over time based on how much light comes back out again at each wavelength. But we cannot really quantify how much blood there is. On the other hand, you can quantify tissue saturation a bit more accurately than some of the assumptions that continuous wave systems do make. We can quantify how much blood is in a given region. And if we combine oxy and deoxyhemoglobin concentration with cerebral blood flow, we can measure metabolism, or specifically the cerebral metabolic rate of oxygen. And so I'm going to introduce one more instrument, which is called DCS, diffuse correlation spectroscopy. I sometimes like to say it's NIRS 2.0 or 3.0. But what it measures is not just how much light comes out again. In this case, we're using a laser source. It's a long coherence laser. And we don't just ask how much light comes back out again, but how is the light property, in this case, the intensity autocorrelation function, changing over time. And you can do some math behind this. What it gets you is temporal changes of cerebral blood flow over time. I guess that's implied by temporal. So what do the signals look like? We get squiggly lines over time. The bottom one here is a subject that I asked to do paced breathing. So you see every inhalation, exhalation. The tiny squiggles on top here is the cardiac pulsation. On top, that's during a functional task, in this case, putting a probe on the motor cortex, asking subjects to tap their fingers, and you can really see this metabolic demand and functional activation signals. And so just an example of time series that we typically measure. But as I just putting this pictorially of what I said is, we in the lab, we use a lot near-infrared spectroscopy. Let me use a laser pointer. We use a lot of near-infrared spectroscopy systems, commercial ones. We build our own as well. We build diffuse correlation spectroscopy systems, and those two optical instruments combined give us a plethora of parameters. So that's oxygenation, tissue oxygenation, and oxyhemoglobin, deoxyhemoglobin, blood flow, metabolism, and whatnot. So I'm going to introduce you to one more thing before moving into how do we actually use all of this to make clinical, to correlate with clinical outcomes and for patient monitoring. And so specifically, what we're interested in is how does the brain, how much perfusion, roughly speaking, is there, and how does metabolism go hand in hand with neuronal function? So we care a lot about neurovascular coupling. And there specifically, there's a connection between neurons and the vasculature, and that's called neurovascular coupling. And so I can draw this out as a cartoon and say whenever the neurons are active, somehow the vasculature is going to change. So that's fMRI signals, near-infrared spectroscopy signals, and the connection between them is mathematically described by a convolution product. Detail is really not important, but the idea is that there's a connection between them, and we can write an equation that says, given that neurons are active, what does the hemodynamics look like? And I can also ask questions about, does this coupling between them change as a function of disease? And so just to say we can measure neurons, in this case I'm showing you a visual evoked potential with EEG. We can measure the hemodynamics with NIRS. So that's just pictorially what it looks like. And so before I go into more disease relevant, neurocritical care relevant problems, let me just say, from a research perspective, we do things that look like this. This is not necessarily clinical bedside compatible, but we can buy instruments that have a lot, like 76 channels, optical signals, and 64 electrodes in this case, or for EEG is what we did. We combined the two and said, well, what is the spatial resolution that we can achieve? So in this case, We had a visual task. So we showed a flickering visual checkerboard that a subject was looking at. So that evokes functional activation in the visual cortex. We said, well, how well can we reconstruct the area of activation in the visual cortex? And so what we did is we did this 3D reconstruction, and so the red blobs here indicate it's a group average of 16 subjects. This blob here indicates functional activation in the left and right hemisphere for this particular visual checkerboard. Now, EEG source localization tends to be relatively low resolution, and a lot of people work on optimizing this, so I am not going to say anything bad about EEG reconstruction, but generally EEG source localization, even for epilepsy or whatnot, is much lower resolution. And so if I just do EEG reconstruction, you see a really wide area that is being reconstructed. And if I combine the two instruments and I say, let me use the information that we gain optically to do better reconstruction in EEG, you see a much better localization of where functional activation occurred. And so I'm bringing this up because there's a lot of power to be combining hemodynamic signals as well as neural sources and ask what kind of information can we get out of the two instruments. And so what we really care about, though, is trying to figure out how can we use the next generation of tools, optical tools, or multimodal imaging, sensing for neurocritical care settings. That's really what the lab mostly focuses on. But I would do my lab a disfavor if I didn't also mention that we do a whole lot of other things as well. We're actually trying to make instruments robust enough for human free divers. So that's really waterproof instruments. We're trying to go much deeper into the brain as well, which funny enough has led us to work with marine mammals as well, trying to see can we do deep brain sensing in dolphins as well. And so again, mostly focusing on clinical translation. And for the most part, we're asking, there's a clinical need, how can we develop better tools for patient monitoring? And just to give you a few brief examples, I always say, well, everything we do comes back to pulse oximetry and we scale it up. Near-infrared spectroscopy is nothing else but fancy pulse oximetry. So we're trying to do transabdominal fetal pulse oximetry. We do a lot of brain imaging with optics. We go into extreme environments, such as for human free diving and then clinical translation. And so since I mentioned pulse oximetry, let me also just say we do work on pulse oximetry, and we say, well, pulse oximetry is actually very much known to be not quantitative at low oxygen saturation. So anything below 80% is not really quantitative. And we just ask, can we do better? And it boils down to, we came up with an algorithm that ultimately showed that we can be much more quantitative, even at very low saturation. So that's a human freediver, an expert freediver, in this case going down to 60% SpO2, and we could really still quantify the low saturation. And we found that on average, we have a much lower error than conventional pulse oximetry. Another one that we're working on is blood pressure sensing without needing pulsatility. And so what we do is we place a near sensor on the forearm, occlude the arm to above arterial oxygen saturation, above arterial pressure, and we have found that we are equally with acceptable error. We've gotten the error down now already, but we can also quantify blood pressure, systolic, diastolic blood pressure quite well with an optical sensor. And so I'm just giving you some flavors of the things that the lab is doing. So for instance, we also built oxygen sensors, for pulmonary assist devices, where we're measuring oxygen saturation as well as hematocrit optically through a blood-filled tube. So in this case, on the inlet and outlet of an oxygenator, so very much related to also ECMO settings. And we have shown that we're as quantitative as commercial devices, with the benefit that our instrument is really lightweight and small and portable. And then I said we're focusing a lot on cerebral applications. That's data I've just shown you. Okay, but since Dick invited me, and that's more neurocritical care related, it's a neurocritical care related talk, let me focus a little bit about how we view traumatic brain injury, neurocritical care settings in general. And so for quite a few years now, we have focused on trying to understand how can we use our non-invasive tools to quantify cerebral autoregulation and the relationship to neurovascular coupling, and how can we come up with biomarkers, sensors that are relevant for non-invasive sensing, or how can we create non-invasive bedside compatible instruments in the neurocritical care setting. And so the example that we've worked on probably the longest is traumatic brain injury, broadly speaking, or rather what we're focused on is cerebral autoregulation, like as this classical view of Lassen's autoregulation curve, saying what is the relationship between cerebral perfusion pressure and cerebral blood flow in the context, or what is the relationship to neurovascular coupling, to neuronal function, and how can our systems assist in identifying the state of autoregulation and cerebral function and health in general. And so specifically within there, for the last decade, we've focused on intracranial pressure. And so we said, intracranial pressure, I still believe, is absolutely the missing vital sign, and we've tried to overcome the invasiveness of the current methods by placing an optical sensor on the forehead, measuring hemodynamic signals, and back calculating what intracranial pressure is. And there's quite a few different approaches out there. So this is really just one that we have pursued and turned out to be quite accurate, at least based on the studies we've conducted so far. So let me walk you through the pipeline, and happy to answer more questions about this topic later on. But the general idea is place an optical sensor. That's a nearest device. The requirement is that it cannot be one of the clinical devices right now, because usually the sampling frequency is not high enough, because particularly what we're measuring is ultimately the cardiac waveform. So specifically we measure the PPG waveform, or rather total hemoglobin or blood flow cardiac waveform. And we say that the shape of that waveform changes with intracranial pressure. And so we characterize the waveform itself, have a bunch of different features, use machine learning, in this case, a regression forest tree algorithm, back calculate then with that intracranial pressure. And so the details are all in the papers here. But it's the general pipeline of how we do the intracranial pressure sensing. And we started with preclinical models, and we've moved into the clinic. And so the data that we've collected a few years ago was the clinical translation of this was in the pediatric ICU, where we had inclusion was anywhere from 1 month old to 17 years. Most of them had an EVD. And we replaced the probe on the forehead. So that was again the research grade device. So that's the DCS device that we built. We also had a commercial NIRS device down here, one of the fancier ones on the left hand side. that I showed you earlier. We had 18 patients, and then we extracted the cardiac waveform, applied the machine learning algorithm to it, and then we compared invasive versus non-invasive intracranial pressure. And so the data I'm about to show you, every single color represents a patient. Every dot is a 1-minute estimate. And what we found is that we have a really strong correlation between non-invasive and invasive intracranial pressure. And so the Bland-Altman plot even showed that we're below 2 millimeter mercury accuracy. Now, again, this is a machine learning based algorithm. We used all 15 subjects, 50 to 80% of the data for training the algorithm, cross-validated. So all of this was kosher. But at the same time, it is based on a very small sample size. So the typical question I'm getting is like, how accurate are we for the 16th patient? And so there, right now, we're in the order of 7 millimeter mercury accurate. So it's a question of more data. And one thing I want to point out is that in order for this algorithm to work, we did need arterial blood pressure as well. And it will become relevant later, but blood pressure was definitely a significant contributor to the feature selection. And then it was mostly the first peak of our blood flow or total hemoglobin waveform. But what we also realized is that maybe if we build our own devices that are sitting on a hospital card and require a 4th-year PhD student to operate, that's not the best approach for clinical translation. And so we moved away from that and we said, okay, well, what is the most simple device we can use? And as I said, there are commercially devices already out there that are small, portable, as a button press, and they spit out data that is as good as the instruments that we have developed at CMU, or at least in terms of some signal fidelity. And so we said, like, well, what if we don't use DCS, blood flow changes, but NIRS signals, NIRS 2.0, with total hemoglobin PPG waveforms? Is the algorithm applicable? And what I'm showing you here is we get almost equal accuracy of invasive versus non-invasive. So that's the approach we're taking right now, and we're still trying to scale up and get more data from various different hospitals. Also, the data was collected on children, even though a 17-year-old head is very similar or is ultimately an adult head. There's some optical considerations of why we want to have a much broader adult range to train the algorithm. So that's one example of clinical translation of our optical methods. This non-invasive ICP, again, has been a story for almost a decade at this point. So let me move on and show sort of the most exciting part, I think, is then the connection to what the cerebral perfusion pressure do to neuronal function. And so again, we said, what is the relationship between cerebral perfusion pressure, intracranial pressure, neuronal function? And so to bring back this example here, it's this question of what is the relationship between neural and hemodynamic signals in the context of intracranial pressure and perfusion pressure. And so this was a preclinical study where we had a large animal model where we had cerebral blood flow with DCS, we had NIRS for oxy-deoxy related signals, and we had EEG signal, EEG recording for neural responses. And we gradually increased or decreased intracranial pressure anywhere from 0 to, I believe, 50 millimeter mercury at times. And we did that by putting a cannula in the ventricle. So it was all saline based, and just by raising or lowering the saline reservoir bucket, we could change intracranial pressure. And so that had then influences on cerebral perfusion pressure. So we anesthetized the animals with fentanyl, not isoflurane. So we did see quite a few fluctuations in blood pressure as well, and therefore in cerebral perfusion pressure. And so the question we asked was, if we go a step back and say, well, typically, even from a clinical perspective, we ideally want to treat patients to be in an optimum cerebral perfusion pressure range. What happens to neurovascular coupling outside the range, so at the lower limit and the upper limit of autoregulation? And so in this case, we had a visual stimulus that was presented for 5 seconds, 10 seconds off, to those animals at every stage of, at every level of intracranial pressure. And then we said, okay, so if we move all the way to the upper limit or also lower limit of autoregulation, how does the neural response and the hemodynamic response look like as a function of cerebral perfusion pressure? So just to show you, this is our actually calculated or measured Lasson's curve in those animals. So you really nicely see a plateau between about 60 to 100 millimeter mercury. And then what I'm going to show you here is the visual evoked potential, the VEP, and the hemodynamic response to the visual stimulus. So this is functional activation that we induced, color-coded with cerebral perfusion pressure. And so if we start out with 70 millimeter mercury, the VEP looks quite normal. And just visually, you do see qualitatively quite some significant changes with the VEP that are related to perfusion pressure. And you see the same thing in the nearest measured hemodynamic response. But what I really want to know is not so much about how do neurons changing per se, but what's the coupling between the two. And so we calculated this function, this hemodynamic response function, and we asked, is it different dependent of if autoregulation is maintained or not? And so just visually, you see that the coupling that's indicated by this function here is different depending on if autoregulation is intact or impaired. And so for us, this was really, really interesting because it's given that our animal model was that we induced saline volume in the ventricles, sometimes a CPP of 80 millimeter mercury, which indicates intact blood flow, intact neurovascular coupling, could have been at an ICP level of 30 or 40 because blood pressure went up significantly. We really saw the strongest relationship with CPP. ICP was not actually a good indicator of autoregulation or neurovascular coupling. And then what we also looked at in those animals, so we calculated the metabolic rate of oxygen and asked what is the relationship of CMRO2 and cerebral blood flow, because the relationship or the ratio of the two is very much indicative as well of autoregulation. And so we asked, in the context of perfusion pressure, how does the ratio between them change? And so what we found is there's a significant difference as well between metabolism, so CMRO2 over blood flow as a function of perfusion pressure. So again, that is intact autoregulation, and we saw lower ratios for lower autoregulation or below the lower limit and above the upper limit of autoregulation with significant differences. And putting that again in context with neural function, we also asked, so this is, I should point out, everything that I showed you earlier would require, in a practical sense, the patient to have some form of functional task. And so we really wanted to see what metrics do we have of just naturally fluctuating signals without the patient having to do anything, or in this case, the animal. But so we also asked about just naturally oscillating EEG signals. If we take just the low frequency band, so up to 4 hertz divided by high frequency band, that ratio of low to high frequency EEG as a function of cerebral perfusion pressure also very much showed a relationship with Lawson's curve, so showing a much lower ratio when autoregulation was intact compared to the lower limit and the upper limit of autoregulation. And so kind of shown here in the boxcar as well, saying that at intact autoregulation, the EEG signatures are also very much indicative of autoregulation. And so I like those examples because that does not require any participation. It's really just place EEG probes as well as the optical devices and monitor over time, but those are specific biomarkers we can calculate based on those non-invasive devices. And so the title of my talk, though, is about body-brain behavior. And so far, I've only really talked about cerebral signals, cerebral things that we can measure. What we really care about also is this question of how does blood pressure, heart rate, respiratory rate influence neurovascular coupling, as well as ultimately cognitive function. And so one direction we're going into right now, or the argument we're making, is in order to really understand any form of disease, we do need to understand also healthy fluctuations in the brain as well, especially this interaction of systemic changes and what the effect is on cognitive function and cerebral health in general. And so what we care a lot about is this body-brain interaction. So the idea is how well we function is a function of a lot of different things. There's an interaction between internal states such as drowsiness, arousal, alertness, and whatnot, and cognitive performance. And so the classical examples I like to give is like if you fall asleep behind the wheel, you're going to crash the car. Sleepiness, drowsiness is one of those internal states that will impact cognitive performance. If I'm too nervous with this webinar right now, my heart rate's going to go up and I am not going to be able to speak as fluently. And so there's a whole lot of influence that we already know about. Like if I take a deep breath when I'm nervous, I can lower my heart rate and my performance is going to be better. And so really our goals nowadays are how can we identify and quantify causality between systemic changes as well as internal states such as drowsiness and cognitive performance, and how can we optimize function and behavior given that we understand this relationship between body and brain. And so we just started an initiative at Carnegie Mellon that is actually called Body-Brain Behavior, or B3. And the idea is really to create wearables, health tracking, fitness trackers for the brain, and identify the interaction between body and brain and optimize the behavior. And so this initiative just started a few months ago, but really the vision statement of this ongoing initiative is that we say we want intelligent engineered interfaces for the body, better wearables than already exist even, as well as for the brain, so that we don't need a fourth-year PhD student operating our instruments. And then ultimately also focus on accessibility for all. So that goes hand in hand with scalability, making instruments robust and smaller, to empower individuals to really optimize their health. So think of it as a fitness tracker for the brain. And what we really envision is a closed loop system. So what I really want is ultimately a closed loop system like a self-driving car that has a bunch of different sensors measuring the environment and moving the car in the right direction. I would like that for the brain, and saying like, hey, I want a warning system that says, maybe you should really take a nap right now, because otherwise your performance in this webinar is not going to be as good as it could be. Or in a even more, well, in a healthcare environment, I would really want to have wearables for and real-time feedback loops for brain function that take everything else, the environment as well as systemic changes, into account. So that could mean automatic optimization of ventilator settings to optimize brain perfusion, to optimize neuronal function, to optimize long-term cognitive function. But I do want to have wearables and feedback systems also for military settings, for instance, of warfighters having some sort of knob to tune to optimize attention even when they're feeling drowsy, but also just everyday wearables. As you can tell, I had a whole lot of fun with ChatGPT creating those images, but that's kind of the vision. And so what is needed for this is like, well, we broke it down into 3 parts. We need measurement systems that are scalable and wearable and quantitative for biomarker development. We need the inference, the analysis tools behind it. So that's your machine learning, analytical models, any kind of computational approaches. And the most crucial one is intervention, of trying to understand what knobs do we have to tune to optimize brain function. And so, as I said, the initiative just started. So I'm more presenting you the vision than anything else. But an example here is, think of the non-invasive ICP sensor that I told you about. So we developed the tools that was partially hardware or using commercial instruments. So we can measure the signals, even in a small portable way. We've developed the algorithms behind it, the computational approaches. And I've shown you that there's a relationship between perfusion pressure and neuronal function. What we have not quantified is behavior. And where we're absolutely lacking right now is intervention. And so the idea is, what can I do with the signals, and what other knobs do I have, such as give blood pressure medication or hyperventilation or anything, to not just optimize pressure, but to optimize function and therefore cognitive recovery. And so the initiative right now is a university-centered initiative, so at Carnegie Mellon. And so kind of to again go back to our vision, or rather the mission statement, is at a university settings, we're very good at developing tools. We can test them in animal models, we can test them in healthy volunteers. We're great in also collaborating, although I'm always happy to have more collaborators, but collaborating on clinical translation as well. But really the pipeline we envision is how can we go from basic tool development and basic neuroscience question all the way to this idea of an actual scalable wearable device, where underlying all of this are the amazing computational approaches that people are developing at Carnegie Mellon, and how can we reach that vision. And so our mission statement is very much focused on that. We want to focus on human translation of intelligent engineered interfaces for the body and the brain, and focus on equitability, robustness, and scalability in terms of making tools and systems available to all, really push them out of a university settings into actual translation, which means we're very much interested in collaborating and with industry, as well as create more of an environment for entrepreneurship within the university. Carnegie Mellon is already great at this, but we're going to do much more of that. We're very much focused on this cross-disciplinary collaboration with community engagement, as well as industry engagement. And I think since we are a university, there's also a huge benefit for our students. So there is an educational component here where we really are going to try to train domain specialists who think cross-disciplinary and also create opportunities for them for internship or learning from industry. But ultimately, I'd be happy if this initiative leads to quite a few instruments that can be commercialized and make it all the way through the FDA and find their way into clinical practice. So with that, I do want to thank my lab, all the funding sources, Carnegie Mellon, and Dick for the invitation. Thank you very much.
Jana, thank you. Every time I hear you, it just blows my mind. I remember we were a couple of years ago, you were just talking about B3 as a concept, and boy, it seems like it's really off the ground right now, and congratulations on that. That's really amazing. I think there are some questions. Can you, I think you can read them, and
Yeah.
then I have a question at the end. So.
Okay. Okay, so the first one is high intersubject variability for oxy and deoxy hemoclothing, especially, so this person found differences in how the biomarkers change with respect to cognitive load, saying it's not quite the same application, but I'm curious as to if there's a similar high interperson variability of nearest biomarkers obtained for patients with a disease state. There is, okay, that's actually an interesting question. Let me rephrase it slightly and say, it's fundamentally a question of, is there an interperson variability? Are we all having the same variability between us? And the answer is absolutely not. Like overall, research studies are always designed, or also clinical studies, is like, let's take a group average, but there's a whole lot of variability between every single individual patient and person. And actually within the B3 initiative, we're very much focused on harnessing the interpersonal variability and saying that this variability is not noise, but is a biomarker of its own. So how my signal fluctuates throughout the day is a signature of how I internally feel. So like arousal drifts, alertness and whatnot. And so we see a lot of variability that we normally just average out, but that's the signal we're after right now. So I do like the question. So there is a lot of variability. There was another one that is a very common question, and I appreciate it. The other question was, which signals from the Artinas device are you using to correlate with ICP? And does it account for skin perfusion? So in this case, yeah, that's a detail, but I didn't really want to confuse the audience. We didn't actually quite use the Artina system in this case. It's a different near system that I didn't want to introduce, but same difference. It's the continuous wave intensity signal converted to oxygenated hemoglobin. And so we used, or total hemoglobin, and we used oxygenated hemoglobin cardiac pulses. So really just a continuous wave signal for training the algorithm. If there's influence from skin perfusion, the answer is, luckily we have not seen a lot of influence. And the reason I can say that is because in the animal studies, we put the probes directly onto the skull. In the human studies, there was the skin in between, and the features we extracted were the same between the two, and the correlation was the same. So there seems to be a unique feature of the brain PPG waveform compared to the skin waveform, and the algorithm differentiated it too. So I did not have to we're not too worried about it, but future steps will include skin measurements on its own to regress out that influence. I believe those are all the questions that I've seen.
I actually have one that I'd like to just run by you, and it has to do with ICP and why we're measuring it. And
Yeah.
you know, I think I did one of the very first ICP measurements at Jefferson Hospital that they ever did there back in the 70s. And I think we monitor ICP just because it's something we can measure, and it became a standard of care. And if I'm correct, the brain doesn't have a pressure sensor in it, and it is not sensitive to pressure per se. It's only sensitive to pressure when pressure affects oxygen delivery or something like that, right? So I'm always confused as to why ICP has been kind of this holy grail, or even the non-invasive measurement of ICP has been a holy grail. When I look at your presentation, it just seems to be the money is in many of these other metrics you're calculating, like CMRO2 and brain oxygen and autoregulation and all that. To me, those are much richer metrics to manage a patient by, rather than ICP. So I don't know, maybe you can comment on that.
I kind of expected you asking me this question. And I actually 100% agree with you. I still think ICP is needed, but not because and that's why I pointed out in some of the data that we have, ICP alone is not a good predictor of any other relationship we found with neuronal activation. But CPP is, which is also CPP perfusion pressure drives a whole lot of then also potentially metabolism and oxygen extraction fraction, and is related to metabolism. So I agree with you that I care much more about how metabolically active is the brain, what's the relationship to blood flow, which is needed, and also for oxygen extraction and that driving neuronal function as well. From a pressure perspective, it is still a question of how much oxygen is delivered to the brain at any given moment. And so that goes hand in hand with CMRO2. And it's a long-winded question of saying, it's like, I still think ICP is very important, but not for ICP rather than for calculation of perfusion pressure, which means the relationship between blood pressure and ICP, this whole concept of optimizing perfusion pressure for metabolism is still something that our data very much supports.
Yeah,
and I think maybe we can do it indirectly and say, well, but we don't actually need ICP per se. We could just use metabolism and blood flow and neuronal function, because they're all very much highly correlated from what we saw with perfusion pressure.
Yeah, I think we're on the same page here. It's just that when you go into an ICU, you know, in a neurointensive care room, you see ICP up on the screen, and you say, okay, I can't go above 22 millimeters of mercury because that's what the threshold is. But you know, we used to see customers in our of our product that would ignore the ICP threshold and just, you know, wait till it affects the brain oxygen. So I think in the future, it seems to me, why wouldn't you have the thing that's on the screen with the threshold is something like CPP, you know, as in like CPP opt, and other metrics would be the things you monitor and have thresholds as opposed to ICP. So anyways, just something.
I agree. I would love to see CMRO2 is a continuous time trace. There's actually so many hurdles still with calculating CMRO2, but I 100% agreed with you, which is actually, I mean, sorry, Dick, I don't know if you want to ask me another question, but there's one that came up in the chat as well.
Yeah, go ahead.
But what are the main challenges that I'm facing in the research? And it's there's a bazillion, but currently in a way, I'm going to give you an answer that is multiple answers. One of the challenges is really how can we make us engineers obsolete? How can we develop instruments that are so easy and robust to use to translate to the clinic and say, I want anyone working with the patient to be able to use our instruments. Now that's typically done more on the commercialization side as well. Like universities are not always uniquely positioned to make instruments user-friendly and robust. But I think there should be a much tighter connection to industry to design instruments and reiterate instruments on the academic research side to enable asking the better questions, very much like Dick was just saying. It's like, do we really need ICP? And I say, well, let me just throw out all of the instruments that we've developed over the years. I want to put them all on a patient, and that's great. I've found a lot of really helpful physicians that are willing to collaborate with us. But given that our instruments are so complicated, we're going to get a handful of patients, and I want to go from 15 patients to 1,500. And so there's still a little bit of a disconnect, and that's the initiative very much as the B3 initiative is pushing towards that of like, how can we make instruments more accessible to also non-engineers and make them more robust? So that's a little tricky there. The other one that I find tricky more on the one of the challenges that we're facing all the time is this idea of also which parameter really to look at, because like you talk to neurocritical care folks and pretty much ICP always comes up, but in our reality, even the definition of CPP can be questioned. And so it's like, how do I rewrite textbooks in a way to convince someone that CPP, the way it was defined, may not be right? I actually think that the way we think about autoregulation is somewhat questionable as well. It's just been established for so long. And so it's similar to the conversation I have with a lot of physicians that when I mention NIRS, is almost like a historic bias already of like, well, we've tried NIRS already 20 years ago. And so trying to convince others that the instruments we have nowadays are just so much better that there are worthwhile opportunities. Yeah, very long-winded question to say it's, yeah, there's a multitude of challenges, but exciting ones and exciting opportunities.
Well, Jana, it's at the top of the hour, and I think we're through all the questions. Wanted to thank you again so much for talking to us today. I would love to have you back periodically and get updates on the amazing progress you're making there in Pittsburgh. It's a very impressive. So thank you very much for being our featured speaker. And to those of you who tuned in, thank you very much. And as always, a special thank you to our small but dedicated group in Europe, giving up their Friday evening to watch our webinars. We really appreciate you. Tell your friends that this will be, the recording of this will be up by next week. So again, Jana, thank you so much. And thank you everybody else.